Okay, so today we are entering a new module. So system biology will span kind of this lecture and the next one and then the next one. So we'll talk about like the ideas that we want to combine multiple omics data together in different ways. Today we're going to look at one of the ways that we explore the systems kind of behavior in biology. So we want to kind of switch gears from so before we talk about omics data as like a snapshot of cells or biology, but we know that biological systems also change over time. So today we're going to look at how we can capture the changes in the system and show them as data or as graph. So today's agenda, we're going to talk about how we think about the systems, especially in biology. We will introduce a little bit about multi-omics data integration, which will be the topic for the next lecture, and biological networks, which will be the topic for the lecture after that. And today we'll focus on how we model the temporal dynamics, like the change in gene expression over time, with some equations. Okay, so the reason why we have to think about biology as a system, because it is a system. Each cell has a lot of components, like genes, proteins, and they have rules, like how the genes interact, like how the protein regulate each other. And everything kind of together from the components and these rules like regulation control the phenotype of the cells. So if we want to understand how the disease arises or how the treatment can change the phenotype of the cells, it's not just about one gene being up or down regulated, it's how the whole interaction can respond to the change. So system biology is the focus of that. So they want to view every system in biology as complex, as dynamics, and not just as a snapshot. And this is really needed because when we look at kind of multicellular organisms, like each cells develop into different tissues. And because every cell starts in the same genome, so they start in the same identical state. But during development, they have to realize that this cell is going to turn into liver. It has to activate liver-specific genes and form liver tissue. Cells that have to form the heart has to express cardiac-specific expression and then develop into heart. So cells have to be able to know where and when to express certain genes and protein. So this is done with two kind of components, like within the cell itself, the epigenetics, like methylation and chromatin folding. These can also change inside the cell, so it makes one cell different from the other. And there is also cell-cell communication. So in this course, we are not going to focus too much on cell-cell communication because that kind of require more complex setting. But here, we just want to explore one situation where cells from the front or from the back of the larva, it has to know that they are going to develop into the head, for the insect, or the tail for the insect. If there is no communication, every cell is going to express genes and protein independently, and you will not have a proper formation of the insect body. So people who were first interested in this type of system from way back, they identify genes that are expressed specifically at the head kind of location of the larva, or at the posterior or the back of the larva. And these genes, when they analyze further, how can cells that are in the embryos know where is head and where is tail? So they found that there has to be some orientation by the follicle cells from the body. So the follicle cells kind of orient the egg cells in one direction. So the egg cell know where it touched the follicle, and that kind of align the head to tail direction. And they found that some of these genes, like decoy or nanos RNA, so these are RNA that can be bound, they are recruited by the cytoskeleton. So it's not just kind of floating around in every cell. It can be captured by the cytoskeleton, by the membrane of the cell, and that kind of localize the RNA toward one direction versus the other. And it's the gradient of these genes that are higher in one direction and low in the other direction that define the head to tail orientation of all the other cells. The other cells sense the gradient, the change in concentration of these RNA. And they realize that if they are high in decoy, then they are located toward the head. If they are low in decoy and they are high in nanos, then they are located toward the tail. So now each cell receives kind of the global signal in the form of gradient, right, for these two genes, these two proteins, and they can realize the next step right, for their development, depending on the expression of decoy and nanos. So this is an example, like a quick example to show that we need to think of gene and protein expression not just as like a number that describes the cell, but in the formation of multicellular organisms, they have specific location and specific time that they need to be expressed. And that's the kind of systems, one of the systems view of biology. So let's look at the approaches that we can use to analyze this kind of system where we have interaction, right, and roles from different genes and different proteins. So the first thing, this will be the focus for the next lecture. So we talked about how we analyze multiple types of omics data together. We learn about genomics. We know that we can identify mutation, identify gene copy numbers. So we know about the state of the gene. Is it wild type or is it mutant? And how many copies there are. We know the state. When we learn about epigenomics, right? DNA methylation or transcription factor binding or histone modification. Now we learn about the chromatin state, like the state of the chromatin. Is it open or closed? Is it active or inactive? And then we learn about transcriptomics. So we know how to measure the activity of the gene in terms of transcript expression. And also isoform. Right? We can distinguish isoform, so we know the activity of splicing as well. When we go through proteomics, now we shift from gene expression to protein expression. And with proteomics, we can also study like protein binding and protein function as well. We are not covering metabolomics and phenomics in this class, but basically with metabolomics, we know that proteins sometimes they act as enzymes that synthesize certain metabolites or process certain metabolites. And if we know like the level of metabolites in the cell, like, sometimes it can tell us about the state, right, of the cell as well. So people who study metabolomics want to know the abundance of each metabolite in the cells to understand what happened. And at the most macro level, like people call this phenomics. So we look at the phenotype of the cells or the sample, and that can describe the state of the sample. It's the topmost level. The other omics can tell us about the molecular information underneath the phenotype. And one thing that we cannot escape, and you will see it everywhere in this lecture and the next one, is every time we want to describe the dynamics or the interaction of genes and proteins like inside the cell so that we understand how they work together and how they change, we always need to draw some kind of graph. So these networks like that describe that protein X1, maybe it binds to X2 and then activate X3 like with certain binding constant. These are diagrams that are drawn as nodes, like vertices and arrows. These are called graph or network. So in the next two lectures, we're going to talk about how we can represent data as networks and how we can analyze data in the form of networks to understand important protein or to understand how the drug treatment change the interaction between genes. And another way that people analyze the dynamics of the system is to look at the change in metabolite. They call this metabolic flux analysis. So basically, we want to understand when we have certain treatment, like, does it reduce certain metabolite or increase certain metabolite, and when the system kind of shift to a new equilibrium, like what is the new stable level of different metabolites. So some people also focus on this as well. But in this lecture, we are going to use one tool. And I'm not sure if all of you have seen this from calculus, but it's a continuation of calculus that you usually study in university. It's called differential equation. It sounds scary, but the idea is, so let's look at some simple example. So let's say we have a protein, and we want to model the dynamics of this protein. So we might say that, okay, this protein is being produced in the cells at a rate of 6 molecules per minute. So it's simple. The cells just keep translating RNA into protein at a rate of 6 molecules per minute. So if we want to write this as an equation, when we say it's produced at a rate of 6 molecules per minute, that means that if you use kind of x here to represent the number of molecules for protein X, and the subscript T here is the time. So the number of protein molecules in the next minute, like T plus 1, it's the next minute. Subtracted the number of protein currently at T minute has to be 6, because this is the production rate for the protein. It's produced at the rate of 6 molecules per minute. So this is just a difference between the number of protein in the next minute versus now. And to write this as a differential equation, which describes the rate of change. So differential equation means equation that describes the difference. So we want to show the rate of change for this protein. So it will be number of molecules in the next time point, like t plus 1, minus the number of molecules in the current time point, xt. And then we want to convert this into rate, so we divide it by the change in time as well. And the change in time here happens to be just t plus 1 minus t, which is 1. And this, in calculus, people will write this as dx by dt, which basically say that it's a delta x, like the change in number of molecules, divided by delta t, the change in time. In the case of 1 unit of time, the denominator happens to be 1, so it's not important, but you can change a unit of time to anything that you want. So basically, we change from this verbal statement, like production at a rate of 6 molecules per minute, into an equation. dx by dt equals 6. So the rate of change is 6 per unit of time, which is 1 minute. If you want to solve this equation, because we know that the protein is produced at a constant rate of 6 molecules, so the number of molecules of protein in the future, like no matter how far in the future, is going to be just 6 times the amount of time, because we produce 6 and 6 and 6. So add up together, it will be 6 multiplied by the amount of time. So the solution for this differential equation, which has a constant production rate, is a linear increase. So this is simple. Look at another example. So we will think about the fact that protein also degrades. In our cells, we destroy proteins all the time so that it doesn't accumulate. So let's say protein X degraded at a rate of 1 in 10 per minute. So the rate of degradation is not a constant, because if you have more protein, if you have more molecules, you have more chance to degrade them. But if you have few protein, like few molecules, then you don't degrade a lot. So the rate of degradation is not going to be a constant. Like we are not losing 2 protein per minute, but we are losing 10% of the protein per minute. So the degradation usually described as percentage. Some percentage of the protein will be degraded. So in this case, the number of proteins in the next step will be 90% of the number of protein that we currently have. So it will be 0.9 times the number of protein from the prior time. If we write again like the difference between protein molecules divided by the difference in time, in this case, it's no longer constant, but it's minus 10% of the amount of protein. So this means that the rate of change of the protein is a decrease, like a minus sign is a decrease, and the decrease rate is 10% of whatever number of molecules of protein that we are having. So the solution for this equation would no longer be linear. it will be now exponential. So we have 0.9 to the power of time. 1 minute you lose 90%. 2 minutes you lose even more, 90% of the rest. And the third minute you lose 90% of the rest again. So the solution becomes exponential. And you are already familiar with this, probably somewhat, like from either calculus or chemistry class. Then we talk about exponential decay, like for radioactive material. Like we saw this graph a lot, where the molecules lose half of the molecule is lost after a fixed time step. And this graph, when you learn in chemistry, like they might tell you that this is a exponential decay, and it's the exponential function with negative power. But if you learn about differential equation from the perspective of calculus, when you have this kind of equation, the change in molecules over time is a minus of certain constant multiplied by the concentration of the number of molecules. This is what we kind of have earlier, like minus 10%. The solution of this is also in the form of exponential, because when you differentiate exponential, you get this solution back. So knowledge from calculus and chemistry agree on the same general form of exponential decay. So we know that we have very simple description of protein expression. One is a constant production, and it gives rise to a linear increase. The other one says about the percentage degradation in percentage, and you get an exponential decay. So in the cells, these two effects are going to work together at the same time. Like some proteins are going to be produced, but some molecules of the protein that already exist will also degrade. So we'll have to combine these two effects together. So let's look at how we handle kind of this situation manually first, and then we can look at more complex situation later. So let's say protein X now inside the cell is being activated, it's being produced at a rate of 1 molecule per minute. But it also degrades. Let's say it's being degraded at a rate of 50% per minute. So we are losing the protein, but we are also producing the protein. So if we write it as a differential equation, it will look like this. The change in protein over change in time is +1, like 1 molecule per minute, and then minus 0.5 of the X, because this is a loss of 50% every time. So we have this equation. We will not worry about kind of solving this the calculus way. We're just going to try to do this like manually, maybe in Excel or on paper. So let's say we start with 4 molecules of protein at the start. So after 1 minute has passed, we got a production of 1, so it's going to be 4 plus 1. But then because these 4 molecules of protein, half of them are also going to be lost due to degradation. So 4 molecules, we lose 2 and we produce 1, so we have 3. And when we have 3 molecules, we lose half, we lose 1.5, and we produce 1, so we get 2.5. So in fact, the number of molecules should be integers, right? But in this case, I'm not going to worry too much. We are just going to add and multiply the number normally and see where it takes us. So if we start with 2.5 molecules of the protein, we lose half, we lose 1.25. When we produce 1, so we get 2.25. If we follow this process, what you are noticing is the number of molecules of the protein is going to continue to reduce, but then slowly from 4 to 3 is minus 1, from 3 to 2.5 is minus 0.5, from here is minus 0.25, minus 0.125, so it's slowing down until eventually it's going to approach 2 molecules. And this will remain 2 molecules throughout. If you continue this process further, you will get 2. Let's look at another situation. Let's say we start with many proteins, 10 proteins. So after first time step, we lose half, we lose 5, we are left with 5. And we produce 1, so we got 6. And with 6 proteins, we lose 3, and then we produce 1, So we have 4. And then the process repeats. Starting from 4, you get 3, 2.5, everything. And eventually you also reach 2 proteins as an equilibrium. So the fact that you start with different number of proteins, but eventually you reach 2 proteins, this is the equilibrium of this differential equation. And what I mean as equilibrium is that if you have 2 proteins, what happens is you have 50% degradation, so you are left with 1 protein, but you also produce 1. So if you approach 2 proteins situation, then the next step will also be 2, the next step will also be 2. So this is called the equilibrium of the system. So inside the cells, basically, if you leave the cells go long enough, this protein will reach 2 molecules and stay there, unless you treat the cell with something that either accelerate the production or reduce the degradation. Otherwise, the cell will maintain the expression level of 2 molecules for this protein, which is a nice property. Like in the cells, you don't want the expression level to change all the time. You want it to stabilize once the cell mature or differentiate. So just a tiny details in differential equation when we do this kind of simulation where we add and subtract numbers, the choice of one step, one time step matter. In the earlier example, we use 1 step for 1 minute, and we lose half 50% of the protein, and we produce 1 protein. Let's say we are being lazy. And we say that I'm going to calculate this per 2 minutes instead. So per 2 minutes, you're going to create 2 proteins, plus 1 and plus 1. And the degradation, because you use 50% for each minute, over 2 minutes, you are going to lose 3 in 4. You are going to lose 75%. You lose 1/2, and then you lose 1/2 of 1/2. So if you write this out as a differential equation, it's going to look different. It's going to be 2, because we produce 2 molecules per 2 minutes. And we're going to lose 75% of the protein per 2 minutes. Even though these seem to describe the same system, but at 2 different kind of time scales. If you perform the numerical calculation, you will reach a different equilibrium. So in differential equation, whenever we want to simulate the behavior of the system, the more resolution we use, like the smaller time step, it will give us a more accurate estimate. So with the time step of 2 minutes, you will get 2.66 as your equilibrium. But in fact, the equilibrium should be closer to 2, which you will get if you use 1 minute in your calculation. So we need to be careful with this, just to show the sensitivity of the model against the unit of time. But in this class, we are not going to worry about these details. We're going to assume that the time unit that we are using is small enough that the behavior that we are seeing is the actual behavior of the system. So let's try to create these differential equations that describe realistic cells maybe for some gene or some protein. So let's start with what you are most familiar with. I'm pretty sure you have seen the enzymatic reaction before. You have enzyme and substrate. The enzyme and substrate can bind to each other into a complex, and then the enzyme can digest the substrate to create the product. So the first part of the equation is reversible, like the binding between enzyme and substrate. They can separate from each other. But the enzymatic reaction where you change the substrate into a product, this is not reversible. Let's assume that this is the situation. In chemistry, we have the reaction constant, like the K on, which is the binding constant, and K off, which is the dissociation constant, and then we have the K for the reaction. And in chemistry, we remember that we can write the rate of change in the concentration for the enzyme, for the substrate, or for the product, or for the enzyme substrate complex. We can monitor the change in concentration for these molecules, these unique molecules. And we learned that if we want to monitor the rate of change in substrate, for example, how the substrate change, it's going to be reduced based on the formation of enzyme substrate complex. Because when enzyme and substrate form, it turns from a substrate into an enzyme substrate complex. So it's reduce the number of reduce the concentration of pure substrate by this term, K binding, multiplied by the concentration of enzyme multiplied by the concentration of substrate. And because some enzyme substrate complex can separate and turns back into enzyme and substrate separately. So we have the rate of change also have a plus term, plus K dissociation times the concentration of the complex. So we have seen these kind of equations before. So one thing that we want to talk about so that we can build more equation is why do these rate of change, why do these formulas take these linear constant, the dissociation or binding constant or reaction constant times the concentration. Why do they take this form? Can we use this also to describe gene expression? And a mental image that I always use to explain this concept is when we think about proteins or genes, when they bind to each other or when they bind to the DNA, because these are 3D objects, these exist in the nucleus in 3D space. They have to, if they were to be able to bind to each other and maybe create some reaction, first they have to run into each other. If they are far apart, there is no binding, there is no reaction. So every reaction that we are going to model, we can think of it as a two-step process. There must be some collision first. The molecules have to fight each other first, and then now that they are bound to each other, then there's a chance that they can generate some reaction or do something. So when we think about the rate of collision, how likely it is for a molecule of the enzyme to meet a molecule of the substrate in 3D space. The probability that they are going to collide with each other scale with the concentration. Because if you have twice more enzyme, like you have twice the number of molecules for the enzyme, you have twice the chance to see that enzyme next to the substrate. So the probability of collision scale with concentration, scale with the number of molecules in the system. And once they run into each other, there is a chance that they will just bounce off or they will bind into a stable complex. And that is a constant, that's the binding constant. So that's why the formation of enzyme substrate complex is in the form of concentration of enzyme multiplied by the concentration of substrate. So that term represents the probability that the enzyme and the substrate molecules will meet each other, will collide with each other. And then multiplied by binding constant or the association constant. So these two factors, these two physical processes together give us this formula that the rate of formation of the complex have to be this form. So we're going to use this idea to build models that evolve around DNA, transcription factor binding to DNA, RNA polymerase binding to DNA, and then the transcription create new molecules of RNA. So for example, we think about transcription factor being bound to DNA as one reaction. It's a reversible reaction. Transcription factor can be released from the DNA. And when the transcription factor is bound to the DNA, it can help recruit the RNA polymerase. So this will be another reaction. So RNA polymerase, which are free, meets with the transcription factor bound DNA, and it got recruited onto the DNA. So it became a single complex with the DNA. And the transcription can be modeled as this RNA polymerase bound to the DNA create the RNA molecules, but the RNA polymerase is also released from the DNA. So these are reactions that we can think about when we model the process of transcription as a 3-step process, which will make the problem too complex. So for the sake of simplicity, in this class, we are going to assume that we will model transcription as a 1-step process. We just have DNA and RNA polymerase bound to the DNA at certain probability, and then we generate the RNA from it. And the RNA can degrade, so there is some degradation constant. And when we have these two effects, like the production with a certain rate, right, and the degradation as a percentage, so the differential equation for RNA expression is going to be similar to the example that we saw before. It's going to be some production rate, a constant, subtracted by a certain percentage of the molecules that will be degrading over time. And when we combine these two effects, the graph will look like this. So the constant production, we know that it's going to give rise to a linear increase, like a line. But the degradation as percentage is going to give us a exponential decay. So when you take the linear production and then subtracted by the exponential decay, you get something that looks like this. So it starts off like as similar to a line, but the slope of the line continues to decrease until it became 0. And this is at the equilibrium where more RNA are being produced at the same rate of RNA being degraded. So you have equal production and degradation. So basically, it's the situation where this formula is equal to 0. You have production in green, K transcription, equals to the degradation times the current concentration. These two cancel out, you get the equilibrium. So it's similar to when we want to find the where the slope of the graph is 0, like we take the derivative and set it to 0. So in the case of differential equation for the dynamics of gene expression, if we want to know like the equilibrium, like the level of RNA that will be stable over time, we have to set this expression to 0 and then solve for the concentration for the RNA. Okay. So let's look at this process, like maybe in slightly more details, like with more numbers, just to get a sense of what happens when we draw this graph, like what happens inside the calculation. How do we derive this? Okay. So this is when we describe differential equation, like we say that the number of molecules for the protein, like represented by x here, like, and t is the time. So if we want to know the number of protein in the next time, like the next time step, is the current number of protein and then plus the difference. The difference is just next time step subtracted by the current time. And to tie this to the differential equation, we divided the difference in x by the difference in t, the time. And this, if you recall calculus, like when we measure the delta x like by delta t, like by the change in time, sometimes we call the x part as the first derivative. But we are not taking any derivative here, we're just representing this quantity in a more compact form. So if we want to know like the number of molecules in the next time step is the current molecules in the current time step, like what we have right now, plus the change, like the rate of change. Okay, so, and when the system that we're going to see from now on, we're going to describe this rate of change. We're going to describe how the protein is created and destroyed. And then we want to use this information to draw out the number of molecules at each time step. So let's see, we have this kind of form from previously. Let's say we have a certain number of molecules at the start. Let's say the cell is in a certain state, and they have this many gene or protein molecules. And this is a description of how the number of molecules change. So if we want the number of molecules in the next time step, it's going to be the current number, which is x0, and then plus the change in number of molecules, which will be the transcription rate, which is constant, and then minus the degradation rate, which is a percentage of the number of molecules. And we can keep doing this. When we want the number of molecules after two steps, after we get x1, we use x1 to plug in, and we get x2 from the same formula. So you will see how this can be done recursively. You can keep doing this to get the next step. Okay, let's do this with actual numbers, so it's easier to understand. So let's say we have a certain gene that being produced at a factor of 4 molecules per second or per time unit. And we have a degradation of minus 20%. So 1 in 5 molecules will be lost. So let's say we start with 100 molecules of this RNA. After one step, we produce 4, but we lose 20 due to degradation. So we're going to be left with 84. The next time step, we have 84, and we produce 4, like the cell produce 4, and we lose 20% of 84, which is about 70. So we're going to be left with 71. And the next step, we start with 71, we put the cell produce 4, and we lose 20% of 71, which is about 40. So we have 61. And so on. So we can keep doing this until it's going to approach the equilibrium at the number of molecules being 20. And that's because if you plug in 20 here, 20 times 0.2 is 4, right? And it cancels out the production. So the number of molecules is going to reduce from 100 all the way down to 20, and it will stay there. That's the equilibrium for this gene. If we change the condition, right? If we start with no molecules at all, so in the beginning there will be production of 4, right? And nothing is lost because you don't have any degradation. And then it will keep increasing until eventually it will also reach the 20 molecules because that's the equilibrium. Right? So we now see that this type of model that explains the number of molecules of gene in the cell, right? Wherever you start, it's going to either reduce or increase until it reaches the equilibrium, assuming that we don't do anything to it. And this model, I want to remind that it actually looks like when we talk about single cell trajectory analysis, and we talk about RNA velocity model, that they think about transcription as a two-step process. DNA produce unspliced RNA, and unspliced RNA getting processed into spliced RNA, like mature RNA, which can then be degraded. If we write this out, now that we know about differential equation, right, we can actually derive this description into equation. So we have the rate of change for the unspliced form of the RNA being plus alpha. So this is the transcription rate. It's the production of unspliced form from the DNA. And then it's going to be turned from unspliced into a spliced form with a reaction rate of beta. So the rate of change for unspliced RNA is going to be plus alpha subtracted by beta times the number of molecules, because the splicing operates as a fraction of a variable unspliced molecule. And similarly, we can write the rate of change for the spliced isoform. It's going to be added by the splicing, by the beta times concentration of the unspliced, but some of it is going to be degraded with a rate of gamma. So the equation is going to be the rate of change for the spliced form over time is going to be beta times the unspliced, and then subtracted by gamma times the spliced form. And this two-step process, if you add them together, if you add these two together, on the left-hand side, you have the difference, like the change in unspliced and spliced form together, like the change in total RNA, unspliced and spliced. It's going to be alpha minus gamma times the concentration of the spliced form, because the beta times unspliced here cancel out. So the reason why RNA velocity, when it treats this process as when it treats transcription as a two-step process, it gives rise to dynamics that we don't see before. Right? Because if we compare these two models, so in our earlier example, we imagine that RNA is a one-step process. Right? We have a production and we have a degradation. So we compare these two. Right? On the right-hand side, it's the same thing. Right? We have transcription rate subtracted by degradation rate of the spliced RNA. But in our prior model, right, our simple model, one-step model, this expression describes the change in only the splice form. Like we only think about the splice form, which has the degradation. But as a two-step process, it actually combines both the unspliced and the spliced form. And the unspliced form is kind of hidden from us in the simple model. So we will not see this type of dynamics. If we compare the graph that we saw earlier, going back to many steps. Okay. If we compare this graph, if we compare this graph, like it's a smooth increase and then reaching equilibrium. If we compare this to what we got from the RNA velocity, which is a two-step process. We'll see that the behavior that we saw before is similar to the behavior of the unspliced RNA, but it's different from the behavior of the spliced RNA. And this is because only by dividing this into two steps that you can see that the dynamics for the splice form is actually different from one step model. So this kind of explains. Now we realize that why these two look different. Okay, so let's look at how we can use these to understand real systems in biology. We talked about these hypothetical systems. So let's see how we can look at different ways that transcriptional regulation occur and capture them with differential equation. So let's say we're going to use this model where the gene is going to be activated for a certain period of time, and then we will turn it off. So the graph will, in general, we look at this. We have an activation of the gene, like balancing with the degradation, and then when we turn the gene off, there will only be degradation. So we will see the exponential decay. So we are going to start from this pattern, which has no regulation at all, like just turning on for a certain period of time and then turning off. So let's say we have some regulation from proteins or other genes. So in a negative autoregulation model, we are going to describe a gene that when it's produced, when it transcribed and translated into protein, the protein form comes back, bind to the promoter or bind to the enhancer. And in this case, it's going to deactivate the transcription. So we call this negative autoregulation. It's going to be negatively regulating itself. Right? When we produce a lot of it, this protein is going to come back and turn it off. So if we write this as a differential equation, it's going to look like okay. So in this case, like we have not only the amount of gene X that we need to monitor, we have another parameters and the state, and that is the state for the DNA. So in this case, this protein, if it can bind to the DNA, it's going to turn off the transcription. So the transcription the transcription activity is now is not constant. So in our model before, like transcription is a constant. Like we produce the certain number of molecules per minute. In this case, it's going to be multiplied by the numbers of unbound DNA, because transcription can only occur on DNA that doesn't have protein X. So now the first term have parameters as well, not just a constant. And we have to describe like what's the bound and unbound nature of DNA based on protein X. And we can mimic what we know from chemistry. We have two compounds basically, protein X and DNA being two compounds. And they can bind to each other to form a complex. So in this case, we're going to write in general that protein X might have to assemble n molecules, multiple molecules of protein X might have to assemble into a complex, and then it can bind to the DNA and then prevent transcription. So using this kind of equation, binding equation for the protein X and the DNA, we can write the change in bound and unbound DNA as also as a differential equation. So the rate of change for the bound DNA is going to be in this form. Bound DNA is created when n molecule of X bind to the meet the unbound DNA. And some of the bound DNA can also, some of the protein X can dissociate from the DNA. So there is also the minus term here. So this is also a dynamic between more protein X kind of binding to the DNA and some DNA with protein X already kind of release protein X from itself. So if we want to look at what's the equilibrium, at equilibrium, what's the fraction of bound versus unbound DNA? Basically, we just set this to 0, set this expression to 0, and then solve for the form for the bound and unbound DNA. Because bound and unbound DNA, when you add them together, that's the total number of DNA in the cells, which is a constant. So we can solve this, and we will look at this. You don't have to worry about the mathematics, but if you're curious, if you replace unbound DNA into this equation, set it to 0, and you will get this. This is also known as Hill function, if you are curious. So this formula kind of describes the fraction of active or inactive DNA that can produce, that can transcribe the gene. So if we plug this back into the differential equation for this gene X, we know that the transcription is going to be based on the transcription rate, which is a constant, multiplied by the fraction of DNA that are active, that are not bound, that are unbound. And that fraction will be this, the input. So I use color code to separate out the different components. So the transcription is going to be product of the transcription rate, and the fraction is a fraction, so it's on the denominator. This is a fraction of DNA that's unbound, and this can create, it can still transcribe the gene, minus by the degradation. So the graph is going to look not so different from before. This is without negative autoregulation, so this is the behavior that we saw before. We increase and then equilibrium and then decrease. With negative autoregulation, it looks almost identical. So you might ask why do we have this in nature? Why do some genes or some protein have to regulate itself in a negative fashion? So two things that we notice from these two behavior. First, if you look at the y-axis, so the y-axis is the number of molecules for the RNA at equilibrium. So at equilibrium, without negative autoregulation, if you produce without any feedback, the equilibrium is about 4. For this system, we have 4 RNA molecules in the system. With negative autoregulation, it's going to be lower, right? Because you have a negative feedback. So the equilibrium is at 1.4. So the expression is lower. And we might wonder, so why do we do this? We even lose the number of protein, number of RNA at equilibrium. The key that people believe is why nature developed this kind of negative feedback is on the x-axis. So when cells respond to change, they want to reach the new equilibrium, and they want to reach it quickly so that everything can stabilize. Without negative autoregulation, even though you reach a higher expression level, it takes about 12 steps to reach that equilibrium. Versus with negative autoregulation, even though you are reaching a lower equilibrium, like less number of molecules, but you are reaching it in less than 5 times it. So with negative autoregulation, the cells can adapt to change and change to a new stable state in half the amount of time in this system at least. So negative autoregulation help reach the equilibrium faster because it can decelerate itself. Instead of going shooting to the high expression level, it stops at a lower level in a shorter amount of time. So with this type of modeling, we can explain the benefit of negative autoregulation. Let's look at another example. This one is going to be much more exciting. So let's say we have a positive feedback. So the gene got transcribed, translated into protein, and the protein comes back and then bind to the promoter or enhancer. But in this case, it's going to promote the transcription even further. So we can mimic the behavior from before, but instead of using the unbound DNA being the active one, in this case, bound DNA will be the active one. So this blue term can change a little bit. But it's a similar looking differential equation. So what would be the difference between positive and negative autoregulation? So we might think that, okay, if we produce more X, right, X comes back, bind to the DNA and produce even more X. Should we get more and more expression until it go to infinity? Is that possible? One thing that will keep this system in check is the degradation. Because if you have more and more molecules of the RNA, you have more and more degradation as well. You have more decay. So even though it's positive regulation, if you're producing more and more, eventually you will reach some equilibrium anyway. But in this case, the equilibrium can be more complicated. So we say that if we want to understand the equilibrium of this system, once we are able to write it, we want to set it to 0 and solve for the concentration of X that makes this whole formula equal to 0. In this case, if you write out the term, it's going to be a polynomial with degree of 3. Basically, there's a x to the third x cubed in the formula. And what we learned probably from high school is that if you have a equation with degree 3, with x cubed, you're going to expect 3 solutions. So in this case, one of the solutions is trivial. If your system produces nothing, it will remain producing nothing. So x equal to 0 with no molecules at all. That's one of the equations. That's one of the equilibrium. The other two, if you write it down, I wrote it out for you. The other two are real. When you have equation with degree 3, sometimes you worry that the roots may be imaginary, but in this case, they are all real. So there are 3 concentrations of X of the gene that you have no change in X. The slope, the change in X is 0. The differential equation is equal to 0. One thing that happened in the case of positive autoregulation is even though we seem to have 3 equilibria, 3 concentration of the RNA that makes the differential equation equal to 0, one of them is not stable. And when I say stable or unstable, when we think about the differential equation or the slope of the graph being 0, there are 2 situations. If the graph is convex, the equilibrium is stable because it's the lowest point on the graph. If the graph is a constant, then you have a slope of 0 everywhere. But the other cases, if you have a concave graph, even though at the top here, the rate of change is 0, it's unstable because if you even produce 1 more molecules, it will continue to increase. If you lose even one molecule, you will continue to lose more molecules. So one of the three equilibria for positive autoregulation, one of the three is unstable. So in practice, in real cells, you only see two equilibria. We will not worry about how I kind of prove this, but you will see with the numbers that if you ever do the numerical calculation as we done earlier in this lecture, for the positive autoregulation equation, what you will find will be like this. If your initial condition, you start with very few RNA molecules, you will continue to lose RNA until you reach the equilibrium at zero. Basically, you are not producing anything. But if you have slightly more RNA molecules than that in the blue region, you will continue to produce more because of the positive feedback until the production and degradation equalize at this equilibrium. And if you have more proteins than that, the degradation will win and you will lose more protein until it reaches this equilibrium. So even though there are three kind of equilibria possible, only two is realized in actual cells. So this is a cool kind of property that when we have positive feedback, we can induce two different cells. A cell with low expression will remain in low expression state, and a cell with high expression will remain in high expression state. So this is what I described. If you have a low number of molecules, you have more degradation than production because you don't have enough positive feedback. So with low concentration, you don't have enough positive feedback. You don't produce enough, so the number of molecules die down to zero. If you have enough protein, enough RNA to have a negative feedback, you continue to produce until you reach the equilibrium. And if you have too high the number of molecules, degradation will dominate and you will reduce the number of molecules until the equilibrium. And people are excited with this property because that means the cell can remember which state it is in. If you have a cell in the low state, even if the gene get produced and get transcribed a little bit, after a while the cells will return back to the low state. And if the cells are in high state, even if you have slightly more production, slightly less production, the cell will bounce back to the high state. So that means that if you start with a cell in low state, also, let's say you treat the cell with drug, you treat the cell with certain drug that activates the expression up to here, and you stop giving cell the treatment. If you don't have, if this gene, the gene that your drug can target, if that gene doesn't have this mechanism, once you stop the treatment, the cell will go back to the original state. But if the protein that you are treating, the protein that you treat with drugs, have the positive feedback mechanisms, once you treat the cell with drug, the expression reach up to here, you stop giving the drug. The cells will reach the new equilibrium on its own because that's the positive feedback inside the cell. So that means that we can kind of change cells from one type to another, and we don't need to maintain the perturbation. We stop the perturbation, and the cell will maintain itself in that state. Similarly, if you start with a cell in a high state, and you replace the expression of the gene until it became very low, and you stop the repression, the cell will not bounce back. The cell will remain in the low state. So we call this the way that cells can memorize its own transcriptional state without having to have the stimulus all the time. Okay, let's look at another example that can also create the same type of this kind of memory. In this case, we only use one gene. Right? We have one gene with positive feedback. But in cells, we can have multiple genes working with each other. So people have identified this kind of a genetic switch where one gene, LAC-I, replace the transcription of another gene, TET-R, And TET-R also can replace the transcription of lag I. So it's like a competition between two genes. Lag I can replace TET-R and TET-R can replace lag I. So there are two equilibrium in this system. If you have high expression of lag I, it will replace TET-R, and if TET-R is replaced, nothing will stop lag I from production. So you will maintain high expression of lag I. On the other hand, if you have high TET-R, it will replace lag I. And with no lag I being produced, nothing stops TET-R from being produced. So you continue to have high TET-R. And in kind of in vitro experiment, like people design, identify compound that can inhibit lag I or inhibit TET-R. So people can test that we can kind of shift the cell back and forth by treating the cell with IPTG or ATC and see that the cell switch from high lag I to high TET-R and back and forth. So people confirm that this kind of two-gene system can also change the state of the cells. And because once we activate one of the two genes, that gene will replace the other. So we don't need to maintain the perturbation. Once we treat the cell with IPTG, we replace lag I, TET-R became dominant. We can stop treating IPTG. And TET-R will maintain the system by itself. In terms of simulation, like if we calculate out the numbers, it will look like this. If we start with high lag I will continue to be produced and we will replace TET-R. So TET-R will go lower and lower until we reach the equilibrium. If we give IPTG, like if we treat the system with IPTG, that neutralize lag I. So lag I will stop replacing TET-R. TET-R production will start, and when TET-R production starts, it will replace lag I, and the dynamics will switch to a different state. And we don't need to maintain IPTG throughout. We just treat IPTG for a certain period of time until TET-R wins over lag I, and then the cells will reach the equilibrium by itself. So people found that in nature, when we have multiple genes working together, cells can generate different equilibrium where it can reach. We can respond to treatment in a different way. And by modeling these with differential equation, we can visualize, we can understand the effect that goes on inside the cell by looking at the behavior as a graph like this. So I would like to end with another system that's also very interesting. So we want to keep things simple. So in this case, we're going to also examine two genes, but we are not going to make the relationship between them very complex. We're going to say that these two genes either activate or replace each other in a linear fashion. So these K's are constant. You can pick 4 numbers for these different K. So this K11 is the positive or negative autoregulation of X1 onto itself. If K11 is positive, that means X1 activates itself, like it's a positive feedback. If K11 is minus, like it's a negative, this means this is a negative feedback onto itself. And K12 and K21, this is how gene X1 and X2 replace or activate each other. So you have many choices. Like you can pick any numbers to replace these 4 concepts. And in the next assignment, one cool thing that you are going to see is, depending on how you pick these concepts, you can induce many kinds of dynamics inside the cells. If you have a cells with 2 genes, like very simple cells, and the mechanism of regulation is very simple as well, like just positive, negative regulation as a linear, or activate or replace each other as a linear. Just these 4 numbers, you can create systems where the cells will die. Nothing will get produced. Both the genes will replace each other until nothing gets produced. or you have an explosion. Both the genes activate each other until you have very high expression. Or you can have a continued fighting back and forth between the two genes until you reach an equilibrium. Or the two genes will continue to fight each other, like one replace the other. But it does so in a way that you will have even more and more fluctuation until the end. So the purpose of this class, as a start of the system biology module, is to show you that by thinking about biological object like biological system, like cells, like as a dynamic process, we can understand the mechanism inside. We can capture different behavior that the biological system can behave with these kinds of simple simulation. We don't make a lot of assumptions here, like we only look at one or two genes at a time, and we can still identify many surprising phenomena inside the cell. So just to summarize, so biological system consists of components, genes, proteins, and also rules of how they interact with each other, like which gene activate or replace the other gene. So that's one layer of the dynamics of the system. You have interaction between components. The second layer is time. When one gene changes, the other changes, and over time, how do they behave with each other? And this can be described with differential equation. And the property of a system, in order to understand the property of a system, we need to understand the interaction between multiple genes and multiple proteins. For example, in the negative autoregulation example, we saw that it reduces the equilibrium expression level, which looks bad, but in exchange, it gives a better response time to the cells. Cells reach equilibrium faster. So if we don't analyze the system as a dynamics over time, like we don't see this property. We only see the expression level change, but we don't notice about the time it takes for the expression level to change. And just to remind, so in the next lecture, we're going to talk about how we combine multiple omics data together so that we understand the biological system from different kind of layers, like from mutation, compared with epigenetics, compared with gene expression. And after that, we're going to talk about networks, like how we draw graphs, how we draw interaction between genes, between proteins, and how can we analyze that kind of data. Okay, so that's everything I want to talk about today.